So nucleotide degradation is where nucleotides get metabolized and broken down in the body. So for the purines, here we're talking about adenine and guanine. They generally get broken down into a substance known as uric acid. So uric acid is associated, its buildup and crystallization is associated with the disease known as gout. Gout was, in the olden times, usually affiliated with individuals that were very rich, ate a lot of meat, drank a lot of wine, but this uric acid tends to crystallize in the extremities of the body. Now we know that that uric acid crystallization is associated with the disease of the excessive breakdown of adenine and guanine. Basically, we're talking about how nucleotides get broken down and metabolized in the body, whereas previously, in the previous lectures, we were focusing on the de novo biosynthesis. This uric acid can further be metabolized, and then we also want to round up our discussion talking about the pyrimidines. So the pyrimidines, if you may recall, are your cytosine, uracil, and thymine. They generally get degraded to their coenzyme derivatives for further breakdown. So in a summary of nucleotide metabolism, you have to remember that ribose 5-phosphate through de novo biosynthesis gets converted to the purines AMP and GMP. That ribose 5-phosphate also can get converted to the pyrimidines, which were UMP. From UMP, we go to UTP. From UTP to CTP, glutamine is the amino group donor to generate CTP. From UMP, we actually have to go to UDP, and then UDP gets converted to deoxy-UDP. That enzyme is ribonucleotide reductase. Then we have to take a step back. The deoxy-UDP gets converted to deoxy-UMP, which then gets converted to deoxy-TMP. Thymidylate synthase does that reaction. Tetrahydrofolate is the donor group of the methyl. So the pathway for de novo synthesis for pyrimidines and purines were studied in a previous lecture. Now we're exclusively focusing on their breakdown products. Purines are going to get broken down to uric acid. The ribose 5-phosphate actually can get recycled back into the cell. The pyrimidines get broken down to malonyl-CoA. Usually that malonyl-CoA gets converted to succinyl-CoA. We saw succinyl-CoA before in the TCA cycle. So let's first talk about the purines. We're talking about adenine and guanine here. So it's important to differentiate the different types of purines. So in this diagram, we have the nucleotide version. We have the phosphate, ribose sugar, and then the entire nitrogenous base. The nitrogenous base for this particular one is adenine. For this particular one, the nitrogenous base is IMP, or hypoxanthine. For this one, it's xanthine, and for this one, it's guanine, of which AMP and GMP we know come from DNA or go into DNA, or they could be used as energy in the form of ATP and GTP. Now, the first step in breaking down, or catabolism, the opposite of this is anabolism, but we're talking about catabolism, the breaking down of these nucleotides. The first thing that's actually done is we remove the phosphate. So when we remove the phosphate, and it's done in all of these initial primary steps, the abstraction or the removal of the phosphate. When that happens, we get the corresponding ribose with the nitrogenous base. Okay, so these have the sugars and the nitrogenous bases, and that usually has the designation of osine, adenosine, inosine, xanthosine, and guanosine. Again, the ribose sugar with the nitrogenous base. Now, going down further in the pathway here, if we remove the ribose sugar, as we see here, let's highlight that in green here. Once we remove, we'll highlight this in green. Once we remove the ribose sugar, we have exclusively the nitrogenous base. And when we have exclusively the nitrogenous base, that's when we get, for inosine's sake, it will be hypoxanthine. For xanthosine's sake, the nitrogenous base is xanthine. And for guanosine's sake, the nitrogenous base is guanine. So we certainly have a three-step pathway here. We remove the phosphate, and then we remove the ribose sugar. Now the nitrogenous base then gets further metabolized. So here the hypoxanthine can get further metabolized to xanthine, and then the xanthine gets really broken down to uric acid. The uric acid is the one that gets built up in gout, or it gets excreted out in the urine. Okay, same thing happens here with guanine. The guanine can get converted to xanthine. Uh, Both of these reactions are catalyzed by xanthine oxidase and guanine deaminase, respectively. So there's a lot of drugs that target xanthine oxidase, particularly because xanthine is the immediate precursor to uric acid and gout. But ultimately, this is a way to get rid of extraneous or excess amounts of purines. And remember, our purines here are adenine and guanine. Want to also give you some brief background on this particular enzyme here, adenosine deaminase. Adenosine deaminase mutations in that enzyme are associated with the disease known as SCID, and also associated with bubble boy disease. So a very famous boy who grew up in the 1970s actually had deficient ADA, adenosine deaminase. So that particular individual, or individuals who lack ADA, adenosine deaminase, are unable or incapable of converting adenosine to inosine. And really what happens is that this whole breakdown of the purine pathway gets halted. Remember, it's just as important to build, but it's also equally important to metabolize and break these down so that they can be salvaged or taken into other parts of the cell or to other pathways. Well, bubble boy disease is a disease. A lot of research went on this particular boy. Grew up in the 1970s, and the adenosine deaminase was deficient. So he basically grew up in a SCID environment, severe combined immunodeficiency syndrome. And in that syndrome, he could not really live in the outside world. So he practically lived in a bubble, hence it's called the bubble boy disease. A lot of ethical quandaries and medical issues were raised by raising this young child in a bubble. And we'll talk more about this as we continue on with the course, particularly about bubble boy disease and its association with this enzyme, adenosine deaminase. But for now, it's an important aspect of purine catabolism, where adenosine is converted to inosine. So to review this slide, the first steps in the breakdown of these nucleotides is to remove the phosphates, as done here. And then we remove the ribose sugar. When we remove the ribose sugar, we liberate the nitrogenous base. It's this nitrogenous base that ultimately gets processed into uric acid, which gets excreted out of the body. The buildup of uric acid, unfortunately, leads to gout because the uric acid can tend to crystallize. So here we see a little bit of keto and enol tautomerization of the uric acid, which generates urate. And all of these are generated from hypoxanthine and xanthine. So remember, hypoxanthine is when inosine gets processed, and xanthine is when XMP, xanthosine monophosphate, or excuse me, xanthosine gets processed. So those two nitrogenous bases, which are purines, can get converted to uric acid. So the uric acid has a tautomer, which is basically the different juxtapositions of the hydrogen proton within the molecule, the enol form or the keto form. You see the alcohol and here you see the ketone form. So the hydrogen can switch places within the molecule. That's not a big issue. The issue is we generate urate. If we protonate that, we get uric acid. So xanthine oxidase is the main enzyme that's implicated in this generation of uric acid. And xanthine oxidase is an enzyme that's predominant in the liver and the small intestine, and it itself is a mini electron transport chain containing a flavine, cofactor, 2 iron, 2 sulfur clusters, and molybdotaurin. So we talked about taurin when we briefly mentioned phenylalanine hydroxylase. This is molybdotaurin, which means it has a molybdenum center. So here's some drugs that actually target gout, and they specifically target xanthine oxidase. So here is hypoxanthine. So if you remember from the previous slide, hypoxanthine can get converted to xanthine, which in turn can get converted to uric acid. It's this buildup of uric acid, which is dangerous in patients that have gout. So xanthine oxidase helps in that transition. So drug targets that sort of inhibit this enzyme or prevent this enzyme from reaching Km and Vmax are allopyranol, and alloxanthine. So both of these look to me, based on their structure, to be competitive inhibitors of xanthine oxidase. And in fact, allopyranol is one of the drugs that is used to treat gout. It's still pretty much currently used. And you can see the structure of this molecule. The substrate very much looks like the cognate substrate is hypoxanthine, which converts hypoxanthine or xanthine to uric acid. So again, talking about the breakdown of your purines, adenine, and guanine, it usually goes through hypoxanthine or xanthine, which eventually gets converted to uric acid for excretion. And here we have two structural analogs, competitive inhibitors of that enzyme, which generates uric acid. One of them is allopyranol, the other one is alloxanthine. So let's talk briefly about the pyrimidines. So once again, the pyrimidines are your cytosine, your uracil, and your thymine, out of which uracil is found in RNA, cytosine and thymine are found in DNA. Cytosine is also found in RNA as well. And the pathway here generally goes through a coenzyme A intermediate. So let's take, for example, CMP. First thing we need to do is get it to the monophosphorylated form. Once again, as before, we strip away the phosphate. When we strip away the phosphate, we have the nitrogenous base attached to the ribose. So again, this is pretty much the same thing that we saw with the situation with the purines. We have the ribose 5-phosphate attached. When we remove the ribose 1-phosphate, we have the actual nitrogenous base. The actual nitrogenous base by itself is uracil, which can be generated from cytidine, or which can be generated from uridine. Once again, we remove the phosphate first, and then we remove the ribose 1-phosphate, or we remove the ribose and the phosphate to liberate the free N2 base, the free nitrogenous base. It's this uracil that can get converted to dihydrouracil. And this is just an oxidation reduction reaction. There you see the chemistry involved where we hydrogenate that part of the ring. We break up the ring here, and then we actually get an alanine, an amino acid in the beta form. So that's the stereochemistry of that is beta alanine. And then we take an aminotransferase and get methylmalonic semialdehyde, and then eventually through coenzyme A, we get methylmalonyl-CoA. So methylmalonyl-CoA is very interesting because methylmalonyl-CoA through a mutase, or it can also be through an epimerase, the they are epimers of each other, can get converted to this molecule here called succinyl-CoA. We saw succinyl-CoA in the TCA cycle. So basically, with the pyrimidine pathway, you get the coenzyme A version of malonyl-CoA, methylmalonyl-CoA that is, and in a vitamin B12 dependent enzyme, we can get succinyl-CoA. The enzyme that does that is methylmalonyl-CoA mutase, also a methylmalonyl-CoA epimerase, because they are epimers, can also do the same enzyme. And what's interesting is that this enzyme is vitamin B12 dependent. This is why we need vitamin B12. But ultimately, this can go to the TCA cycle, but not only the TCA cycle, also we will find out later on in the course, we will see this pathway again in fatty acid metabolism. So right now we're really talking about nucleotide, right? Nucleotide metabolism, purine and purine. In fatty acid metabolism, we will see this again, where methylmalonyl-CoA gets converted to succinyl-CoA. That vitamin B12 enzyme is going to be important later on, and the enzyme is methylmalonyl-CoA mutase. Very interesting court case involving Patricia Stallings in 1991, who was actually put in jail, but was later vindicated. It turned out that her son had a deficiency in methylmalonyl-CoA mutase. She was wrongfully accused of murder. So we'll talk about this very interesting trial that involved this enzyme a little bit later on in the course. Finally, I want to end this with another important part of nucleotide metabolism, and that is the salvage pathways. So the salvage pathways basically is recycling. So if you look at this metabolic scheme here, we already studied this pathway in our previous PowerPoint slides preceding this. We generate hypoxanthine, then from that we go to xanthine, and then to uric acid, which gets excreted. But there's a way to get hypoxanthine and go back to IMP, or guanine and go back to GMP. Now remember, it's important to jog our memory here. Guanine and hypoxanthine, also adenine here, are free nitrogenous bases. So they're just the nitrogenous bases by themselves. And the cell has a pathway, this is called the salvage pathway, where it goes back. It goes back and puts back in the ribose sugar, it puts back in the phosphate, and you generate essentially a nucleotide, right? IMP or GMP. So this going back or reversing back to the monophosphorylated nucleotide, IMP, GMP, or AMP, that's called the salvage pathway. So salvage pathway basically means going back from the free nitrogenous base back to the monophosphorylated nucleotide. So enzymes that are important in that are two of them. One of them is HGPRT for hypoxanthine guanine phosphoribosyl transferase. It takes hypoxanthine, converts it to IMP, again attaching the sugar and the phosphate. Or it could take guanine and converting it to GMP, again transferring the phosphate and the sugar, ribose sugar. There's also adenine, which gets transferred back to AMP using PRPP. The enzyme that does that is APRT, adenine phosphoribosyl transferase. So what's very interesting in this is a disease, a very devastating biochemical disease known as Lesch-Nyhan syndrome. So Lesch-Nyhan syndrome is a topic that we can definitely discuss in your biochemistry research topics or presentation, but it's worth discovering in the next couple of slides and later on in the course some of the pathology associated with this disease. Pathology associated with disease is self-injury and self-harm. So people who lack this gene, HGPRT or APRT, self-mutilate themselves. They bite themselves, they have to be constrained and restrained, they bite their lips, they bite their fingers, they're very violent towards their caregivers. It's a devastating disease which has horrible consequences for quality of life. So once again, talking about the salvage pathways in this disease known as Lesch-Nyhan syndrome. Once again, we studied this part here, IMP, breaking it down to inosine. And then if we remove the ribose, remember this becomes hypoxanthine. The salvage pathway, as shown here, takes that hypoxanthine and regenerates the IMP, or it takes that guanine and regenerates the GMP. Again, salvage pathway, it goes back. It goes back to the IMP state, the GMP state, or the AMP state in the form of the APRT genes. So it's these three genes, HGPRT, APRT, and adenosine deaminase, though this is mostly associated with SCID and bubble boy disease, but it's these three enzymes with primary emphasis on HGPRT and APRT that are associated with the self-mutilation and self-injurious disease known as Lesch-Nyhan syndrome. It affects children, it affects adults. Most of these have to be on wheelchairs and put in some sort of restraint, and they need constant care and constant caregiving. So this is the pathway that's involved in that. It's very important from a biochemical perspective that we have functioning enzymes and we have a functioning salvage pathway so that we can go back once again from the free nitrogenous base back to the AMP, IMP, or the GMP version of the purines.